Density in Arabic. What do you say density in Arabic? Asata. Yeah. Okay. Thank you. Okay. Please listen. When you want to know what is the mass of a macromolecule, you can use these two techniques. Okay. What is the meaning of MS? What is these techniques? Do you know? MS. What is the symbol? It's the symbol of mass spectroscopy. It's a technique. Mass spectroscopy. Mass spectroscopy. Mass spectroscopy. It's a technique. I will explain it in next sections. When you want to know the mass of macromolecules like an enzyme or a protein, you can use mass spectroscopy or electrophoresis. These are two important techniques in biochemistry. What is your major? Are you biochemistry or molecular and cellular biology? You are the students of molecular biology or biochemistry biology? Biochemistry, okay, great. My major is biochemistry too. It's great, big like. Okay, we have, my major is biochemistry too. Are there any students of biology? Or you are all biochemistry? Molecular biology or cellular biology? You are all students of biochemistry. Okay, great. So, just biochemistry, thank you. And so you should know these techniques very good because your major is biochemistry, and these techniques are special for biochemistry. These are specified techniques in biochemistry, and you should know them, you should learn them in laboratory. When you want to know the mass of macromolecules, you can use mass spectroscopy or electrophoresis. When you want to know about the volume or this slash means or, yeah. When you want to know about the volume or the density of a macromolecule or of an enzyme, you can use gel filtration, centrifugation, pulse-field gel electrophoresis. These are some other techniques that we use usually in biology, in biochemistry. When you want to know something about the charge of a macromolecule, the techniques that you can use are these two ones: ion chains, chromatography, or electrophoresis chromatofocusing. When you want to know and you want to have some data about the shape of a macromolecule, you can use these three techniques: mass spectroscopy, chromatography, and electrophoresis. When you want to know something about the energy of a macromolecule, you can use crystallization, centrifugation, spectroscopy, and biocalorimetry. This table is very important for your exam. For example, let me tell you a question. Let me tell you a question. Ask you a question. I will ask you, I have an enzyme and I want to know the charge of this. For example, I have a protein, okay? Protein A, and protein B, okay? I want to know which of them has more molecular mass, okay? Imagine this is the question of your exam. We have two protein A and protein B, and I want to know about their mass, and I want to know which of them has higher molecular mass. Please tell me what technique I can use to know about the mass of these two proteins. You should tell me the suitable technique for know about the mass of these two proteins. Mass spectroscopy and electrophoresis. This can be a question in your exam. Or imagine I tell you that I have an enzyme and I want to know something about its energy, about this activation energy. Imagine activation energy. Tell me what kinds of technique I can do to understand the energy of my exam. Excuse me, of my enzyme. For energy, I want to know something about the energy of my enzyme. Please tell me which techniques are suitable for doing such this work. Yes, one of them is crystallization, centrifugation, spectroscopy, and biocalorimetry. These are some examples of my questions. Or they are easy, yes? Do you agree with me? They are easy. Mr. Mohammed Yahya said it's very easy. So I can complicate my question. If it's very easy, I can complicate it. When you be in, when you are at home and you want to give your exam at home, you can use this table. But when you are in university, and you want to give an exam, it will be a little harder. But when you are at home, yes, it's easy. You can use this table. Don't worry. The other these were physical attributes. اوکی physical attributes یک سری صفات فیزیکی ویژگی های فیزیکی some physical characteristics but now I want to talk about chemical characteristics. Imagine you want to know something about the chemical characteristics. Like what? Composition, molecular structure, covalent bonds, non-covalent bonds, native or denatured structure. Native structure means nature structure. When we have a natural structure, we say it is native. Do you understand what is the meaning of native? طبیعی, natural. But in the nature structure, no. The structure cannot work at all. Clear? Native structure or the nature structure, yes. Native structure means natural structure. The structure that can have activity. But the nature structure, no, cannot have any activity. Solubility and complex formation. When you want to know something and data about the composition, you can use mass spectroscopy. If you want to know something about molecular structure, you can use spectroscopy and crystallization. Crystallization is a great technique, and when we use crystallization, we can know. Have you ever seen the crystallized structure of an enzyme? Crystallized structure of an enzyme. Let me search it and bring some examples to class. Crystallized structure water. Let me bring some beautiful example for you. Just wait. I want to bring the crystallized structure of water. Crystallized structure of water, just please wait. This is the crystallized structure of water, okay? By using the technique crystallization, you can have the crystallized structure of any molecule. This is the and another interesting picture is this. This one. This is the crystallized structure of water. Interesting, huh? Isn't it? By using crystallization techniques, we can crystallize the structure of proteins, nucleic acids, and other, for example, DNA. بلور, yes, بلور. بلور کردن. Yes, بلور. Okay, let's come back to crystal. Crystal is, we'll write crystal like this. Crystal. It's by Y, crystal, but we can write it by Y. When you want to detect covalent bonds, we can use mass spectroscopy, bioinformatics, and electrophorese. When we want to detect non-covalent bonds, we can use chromatography and electrophorese. When we want to have something and some data about native or the nature structure, for example, someone give me a protein and ask me to detect, is this protein nature or no, it's the nature. I can use these techniques until here. Spectroscopy, chromatography, electrophoresis, proteomics. Do you know what is proteomics? Do you know what is proteomics? No? This is a new technique. We cannot, it's technique. In fact, it's a science. It's a new science. It's a new major of biochemistry. And what does it say? Proteomics, for example, imagine we have a cell. We have the cell. This is one cell. We want to know what kinds of protein are present in this cell. When we want to know what kinds of protein are present in this cell, we can use proteomics. Proteomics tell us that the protein profile of a cell. I can use proteomics to know how many and which kinds of proteins are present in this cell. Understand? This is proteomics. Detecting proteins in the cell. The protein profile of a cell. Clear? Okay. Proteomics, chromatography, crystallography, and precipitation. We can use these techniques excuse me, these are for solubility. Until here is for, until here, these four techniques are for understanding that the protein or macromolecule is nature or denature. When we want to, Mr. Ali, when we want to know that or protein or any other macromolecule is nature or denature, we can use spectroscopy, chromatography, electrophoresis, and proteomics. Use it for what? What do you mean, Mr. Ali? About proteomics? Why we use proteomics? You know what are the use of proteomics? کاربردهای پروتئومیکس رو میخواید بدونید؟ Let me tell you some application of proteomics. Application of proteomics. Application of proteomics. Imagine we have a normal cell and we have a normal cell and we have a cancer cell. A cancer cell. Okay? By using proteomics, we can understand which of them is a canceric cell and which of them is not. It's normal. Because the protein profile of cell change in different disease. Change in disease. We can use it for detecting cancer cells, for example. For example, we have a patient. He say, I think I have, for example, prostate cancer. We can use proteomics because the protein profile of a cancer cell, a prostate cancer cell, is different from a normal cell. Okay? And your friend asked native, why we want to know the protein is native or not. In some kinds of research project. Yes, Miss Marve, it's similar to genomics. But in genomics, you want to know the gene profile of a cell. But here we have the protein profile of the cell. Some kinds of research projects, we use proteomics, we want to know the protein is a native or denatured, and etc. They usually are used for research projects, not in medical projects, okay? They are usually used in research projects, not medical projects. And the other one is solubility. When you want to know and detect some data about solubility, we use chromatography, crystallography, and precipitation. Mr. Ali, I don't understand what you mean. بحثی و لیس طبی. Yes. در تحقیق و در طب استفاده نمیشه. Not using طب. I understand. Complex formation. About complex formation. When we want to have some data about complex formation, we can use electrophoresis, mass spectroscopy, and crystallography. These two tables are very important, and be sure that some of your questions will be from these two slides. Okay, let me tell you some important notes. These are some important notes. The first and the most obvious things is that all these molecules are produced in living cells under mild chemical conditions of temperature, pressure, and pH. Biomicromolecules are built up from simpler building blocks molecules by covalent bonds formed usually with the elimination of water. Moreover, biomolecules are continuously synthesized and degrade in cells in a highly regulated manner. It follows from this that many biomolecules are especially sensitive to extremes of temperature and pH. As I told you, temperature and pH are very important, and they can change the structure and some characteristics of molecules, which may present a problem in their handling prior to and during any biophysical analysis. When we want to analyze a biomolecule, we should take care of that molecule, because the molecule should not be denatured. Do you understand? And the last note, since biomolecules result from a long process of biological evolution, during which they have been selected to perform highly specific function, biology, what does it mean? There is a very close relationship between chemical structure and function. This sentence is very important. من نمیفهمم. I don't understand what Mr. Qasim mean. You mean your research? Surgical. Okay, I understand. Thank you. Mr. Qasim, do you understand what I said? Do you understand English? Or do you understand English? Okay. So, please pay attention to this important sentence. There is a relationship between chemical structure and function. That means that even at pH and temperature values under which the molecules may not be destroyed, it may function suboptimally or not at all. When we want to extract or purificate or analyze the protein, something may happen. Something bad may happen to protein or other macromolecules. They may be denatured. So we should be very careful about them, and we should do something that denaturation not happen. Next section, I want to talk about the factors that affect analyte structure and stability. These factors are pH, temperature, and solvent polarity. Let's study about them. How can pH, or how can temperature, or how can solvent polarity affect the analyte structure and its stability? Okay. This book was the book that I sent you in group. Next section, I want to talk about these factors, and they are in this book, page 9. Page 9 of this book. Write down page 9 of this book. You can study it, we can read it, and be ready for our class. Okay? Thank you. If you be ready for class and if you take part in what I said, like today, I will be very happy and I won't be tired. But when just I'm speaking and you don't say anything, it makes me tired. Thanks for your attention. Thanks for your attention. Any question? If you don't have any question, today my teaching has been finished. Yes, I'm happy. Of course, I'm happy with you, and I'm happy to be in your class. And when you say something, and when I say that you are understanding the lessons, I will be more happy too. Nothing? Any question? Please, until Thursday, send me your homework about that 2 pictures. It has 2 scores. I don't have the list of the class. Unfortunately, I don't have the list of class. Would you please write down the name of all the students of class? What's her name, Miss Marve? Next Thursday, 6 days later. Until that time, please send me your homework about that 2 pictures. Would you please, one of you, write me down the list of the name of the students of class. List of the class. The name of all these students, not just some of these students. No, I asked about two pictures. I wanted you to write two paragraphs about two pictures. Let me tell you. I will tell you. Please wait. Thank you, Mr. Mohammed. Thank you. About these two pictures, interpret these two pictures. One of them is this is one, and the other is this, two, number two. For each of these pictures, write down one paragraph. That's what is going to happen in these pictures. Thank you, Miss. Thank you, Miss Noor. Yes, thank you for your working on this project. Thank you. It has 2 scores of your exam. Okay, I wish all of you to be fine. Thanks for the picture is speak about the polar and of course about the delta S. Don't forget delta S, okay? Mr. Ziad, what do you mean? Interpretive. Interpret these two pictures. Interpret. تفسیر کردن. تفسیر. Okay? Again, I want to tell you thanks for your message that were full of kinds. And thanks God, no, I'm better. I feel better, and when I came to your class, I feel better after your class. Okay? No, I feel better, and you gave me energy. You gave me positive energy. Thank you, Mr. Ali. Yes, Mr. Ahmad. Since just write down one paragraph. Thank you, Mr. Varud. Write down one paragraph about this picture. آقای فارس و دوستانی که فارسی متوجه میشید، لطفاً این عکس رو تفسیر کنن دوستاتون، بهشون بگید. اگر کسی متوجه منظور من نشده، عکس رو تفسیر کنن و درباره تأثیر دلتا اس و دلتا جی در این عکس توضیح بدن. Thank you, Miss نور. باشه؟ درباره اتفاقات ترمودینامیک این عکس توضیح بدن. مرسی. بله، به صورت جداگانه آقای فارس. هر عکس جداگانه. عکس اول 1 پاراگراف، عکس دوم هم 1 پاراگراف. لطفاً ممنون میشم ازتون به دوستاتون توضیح بدید. میترسم کسی متوجه نشده باشه. بعضی‌ها شاید متوجه نشده باشن. باشه؟ لطفاً به دوستاتون توضیح بدید. خیلی ممنون ازتون.